The scientists that I'll be focusing on are S.J. Singer and G.L. Nicolson, and these two scientists were the creators of the fluid mosaic model. First, Seymour Jonathan Singer was born on May 23, 1924, and he earned his bachelor's in chemistry from Columbia. He then went on to get a PhD from the Polytechnic Institute of Brooklyn and was then a postdoc fellow for Linus Pauling at the California Institute of Technology. And in 1949, he helped show that sickle cell hemoglobin differed from normal hemoglobin. He was then a chemistry professor at Yale until 1961, when he left Yale to create a new branch of biology at the University of California, San Diego. And he aimed to create a department that combined biology and chemistry in one cohesive unit. Next, Garth L. Nicolson was born on October 1st, 1943, and he got his bachelor's also in chemistry from UCLA. He then got his PhD from the University of California, San Diego, and he worked for S.J. Singer on the fluid mosaic model during this time. After he discovered the model with S.J. Singer, he went on to get his postdoc at the Salk Institute, and he then focused on cancer research for the rest of his career, while he was a professor at various medical schools. So to go over the plasma membrane, it is found in all cells and separates the inside of the cell from the outside environment, as well as organelles in eukaryotic cells. Some of its functions are to separate organelles, separate the cell from its outside environment. It helps in cell-to-cell recognition. It also manages the exchange of molecules with the outside environment, like waste and other materials. It helps maintain electrochemical gradients and many other functions. So before the fluid mosaic model, the model that was widely accepted was called the Davison-Donnelly model. And this was a model that was also known as a sandwich model because it showed the protein sheet surrounding the phospholipid bilayer kind of like a sandwich. And the proteins were just there for structural reasons, not for any other purpose. And the problem with that was that this was not energetically favorable because it didn't maximize the hydrophobic interactions with the phospholipids and the proteins. So then their fluid mosaic model that they helped create, this also included a phospholipid bilayer, but it was a fluid-like state and the molecules could constantly be moving in the two dimensions. And based on thermodynamics, this was able to get the lowest free energy possible, unlike the previous model. This is also amphipathic, like the other model with the phospholipids, but it helps maximize those interactions, like I was saying. And this shows that some of the proteins are embedded in the membrane, unlike with the other previous model. It has the ionic and polar groups facing the water, and the nonpolar groups buried inside the membrane. And they also differentiate between the peripheral and integral proteins in their paper, and they say that the type depends on the size and structure of the proteins. And this was very different than the previous model, because in the previous model there weren't any proteins inside the membrane, just surrounding it. Now, the impacts of their model, it changed the way that the scientists thought about proteins in the membrane, no longer just as structural and passive, but they could work as signalers, pumps, and transporters. It also explained the structure in a way that made sense energetically. The sandwich model did not make sense in terms of free energy because of those hydrophobic interactions, but this one made those interactions less. It also showed that proteins could exist inside and outside the membrane while maximizing hydrophobic interactions. And then it also explained lateral movement in the membrane through their viscous fluid description. So they say that the proteins could constantly be moving through the membrane like it was a fluid. And since then, there have been many updates and new discoveries to their model. And the models that they continue to come up with and grow upon the original fluid mosaic model just become more and more complex. For example, some of the things that have been updated have been lipid rafts and protein and glycoprotein complexes. And these both make the membrane less fluid than previously thought, which prevents lateral movement. And also, another component that makes the membrane less fluid is a cytoskeleton that is bound in the membrane. So, this also plays a role in decreasing lateral movement of some of those integral proteins. So, overall, the membrane is just not as fluid as they originally thought, at least for some of the integral proteins. And so, after his discovery of this with Nicolson, S.J. Singer was given emeritus status at the University of California, San Diego, where he kept teaching and doing research. And he worked in researching the causes and genetics of Alzheimer's for the next 20 years, while he also taught honor seminar courses. And then he died in 2017 at age 92. After his work in the fluid mosaic model, Garth L. Nicolson also went on to become an emeritus professor at the Institute of Molecular Medicine in Huntington Beach, California, as well as at the University of Newcastle in Australia. He has published work in over 700 medical and scientific papers, and he's been the senior editor of 4 different scientific and medical journals. And here are my resources.